Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Advancing Viral Vector Manufacturing with Innovation: The Importance of the Cell Line. I'm Casey Nevins, an editor at BioInsights, and joining me today are Marguerite Campbell and Brian Tomkowitz, who will discuss the importance of the cell line for viral vector manufacturing, a multi-step process that has historically been lengthy and labor-intensive. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions to our speakers using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. I'd also like to draw your attention to the resources on the right-hand side of the screen, where you can find more information on the topics covered today. Now I'd like to introduce our presenters. Marguerite Campbell is an experienced cell and molecular biologist with over 25 years of experience in the biotech and pharmaceutical industries and national labs. In her current role at SK Pharmteco, she leads the upstream processing team and has played an integral role in the cell line development efforts for viral vector manufacturing. Brian Tomkowitz is an accomplished cell and molecular biologist with over 15 years of experience in the biotech and pharmaceutical industries. Currently, he is the Senior Director and Head of the Research and Development at SK Pharmteco, where his research focus is on cell development and viral vector engineering to improve viral vector manufacturing. So, without further ado, I will hand you over to Marguerite to kick us off with the first presentation.
Thank you, Casey, for this opportunity, and thank you for everyone joining the webinar. We're very excited to tell you about our work with the SK Pharmteco cell line. Today's webinar will include an introduction to the SK Pharmteco, a quick background on viral vector production, an overview on the development and evaluation of our cell line, and next steps, innovations of our cell line. At SK Pharmteco, we have 70 years of manufacturing experience with 2,000 team members over 7 global manufacturing sites covering small molecule, cell and gene, and analytical functions. We have a global presence of 7 production sites, 5 R&D centers, and 4 standalone analytical labs. We are on a mission to save lives, and we're striving for a future where advanced therapies are available and affordable for every patient. With that mission in mind, we set out to generate an SK Pharmteco cell line for viral vector production. Starting with the HEK293 cell line, since they are the predominant cells used for viral vector production, our goal was to generate an SK Pharmteco serum-free suspension monoclonal cell line for universal AAV, lentivirus, and adenovirus vector production to support both our business and client needs. Serum-free suspension provides an animal component-free system that is amendable to manufacturing scale to meet clinical and commercial demands. With an in-house SK Pharmteco cell line, we've reduced costs by alleviating licensing fees and royalties associated with production in commercially available cell lines, and by developing our cell line with cost-effective reagents. In addition, we then have the freedom to operate and innovate our cell line. Our approach to generating the serum-free suspension line from the HEK293 adherent line was three-pronged. The path on the left outlines a stepwise reduction in serum, followed by suspension adaptation. Alternatively, the path on the right outlines suspension adaptation, followed by a stepwise reduction in serum. Although all paths were successful, our path forward was the center path with adaptation straight into serum-free suspension completed in multimedia formulations. This was the most direct and expeditious path forward to generate our serum-free suspension pool. The SKPT pool was evaluated against competitor cell lines. On the left, the average doubling time was calculated over 10 passages, comparing the SKPT pool to VPC 2.0 and 2 other commercially available cell lines. The SKPT pool doubling time averaged about 28 hours and is competitive with VPC 2.0 and is faster than the other 2 commercial cell lines. On the right is a comparison of the median AAV titers. Titer was determined by dPCR from 72-hour harvest, and the SKPT pool produced AAV titers that were competitive with VPC 2.0 and higher than both of the other commercial cell lines. With this promising cell culture performance and AAV titers, we moved forward with generating a monoclonal cell line from this SKPT pool. Monoclonal cell lines were generated by single cell printing with verification by whole well imaging. The top panel documents printing of the SKPT pool with identification of a single cell in the cartridge funnel and the identification of the well for 96 well plate deposition. The bottom panel documents outgrowth with days 0 through 12 at high magnification of the whole well image to focus on the region of single cell deposition to growth into a cluster, followed by late whole well images of cell cluster growth, cluster dispersion to well confluency and expansion to shake flask. We generated just under 100 monoclonal cell lines that were banked. All of these cell lines were screened for both AAV and for lentivirus production. And then the top 4 cell lines from those screens were evaluated for adenovirus production. Clone HEK293 SKPT4G9 was selected as the top clone for universal AAV, LV, and ADV vector production. 4G9 was further characterized for cell culture performance and morphology. The top left graph plots 4G9 viability and viable cell density that was monitored over an 11-day terminal batch shake flask process. The top right panel revisits the average doubling time, comparing 4G9 to competitors and the parent pool. The average doubling time of 4G9 was about 27 hours. The bottom panel shows 4G9 under bright field microscopy with single cell suspension morphology and minimal cell clustering. 4G9 was evaluated for AAV production across multiple serotypes in a head-to-head comparison with VPC 2.0. Transfections were at shake flask scale with 72-hour harvest, and AAV titer was determined by dPCR with multiple primer probe sets. The results show that 4G9 in blue is competitive to VPC 2.0 across all 6 AAV serotypes. We evaluated the impact of cis plasmid size on packaging and AAV production. On the left, our cis plasmids tested ranging from 2.1 kb SC AAV plasmids to SS AAV plasmids ranging from 4.2 to 4.5 kb with various promoters and transgene sequences. On the right, the median AAV2 and AAV9 titers are graphed and show that 4G9 is capable of supporting AAV production for both cis GOIs and all plasmid sizes evaluated. 4G9 was then evaluated in 2-liter bioreactors under 2 conditions in duplicate. AAV1 titers were evaluated from 48 and 72-hour harvest by ddPCR. 4G9 produced consistent E11 AAV1 titers across all bioreactors, condition, and harvest times, confirming consistency and scalability from shake flask to bioreactor. AAV9 was generated in 3 cell lines: the SKPT pool, 4G9, and VPC 2.0. Virus was affinity purified, followed by an anion exchange polish. In the AEX chromatograms, a single distinct 260-280 peak is observed for both the SKPT pool and 4G9 cell lines, and several peaks are observed for VPC 2.0. The predominant peak fractions were analyzed by SVAUC to characterize full particle distribution, and results show that 4G9 had high full particle production at 79.3%. The SDS page gel on the right shows comparable stoichiometric incorporation of capsid bands VPC1, 2, and VPC3 for all cell lines. In a head-to-head comparison to evaluate lentivirus production, a CD19 CAR virus was generated in 4G9 in 3 replicates and in the VPC 1.0 cell line. To determine an infectious titer, sub-T1 cells were transduced, and the CD19 CAR T expression was determined by a flow-based assay to calculate transducing units per mil and is graphed with results obtained from P24 antigen-associated ELISA. 4G9 produced TUs at comparable levels to VPC 1.0. 4G9 was compared to commercial cell lines for adenovirus production. Infections were completed at shake flask scale with multiple MOIs with 72-hour harvest. ADV titer was determined by ddPCR and showed that 4G9 produced higher adenovirus titers than both the XB and other commercially available cell lines. Thus far, I've summarized the HEK293 SKPT 4G9 cell line that we generated and its evaluation for universal AAV, lentivirus, and adenovirus vector production. Brian will now summarize our next step innovations with this cell line.
Thank you, Margie. I'd like to talk to you about a little bit of the work that we're doing in addition to the identification and evolution of the 4G9 cell line. We realize that cost in manufacturing is extremely expensive, and having access to a proprietary cell line allows us to control a lot of the levers that drive costs. And the three biggest cost drivers are obviously plasmids, media, and transfection reagent. The ability to leverage our internal technology and use media screening across the diverse suppliers, as well as internal technology for new media feeds, allows us to decrease prices while controlling supply chain. Having access to multiple transfection reagents, GMP ready available, whether they be PEI, effective based, or others, also allows us to control costs. We've developed new technologies in transfection modalities, supplanting the traditional triple transfection with two plasmid-based reagents that allow for similar titers while reducing the overall cost of plasmids. And lastly, the plug-and-play manufacturing and our ability to use this universally in our manufacturing processes shows that we've invested in a platform that's amenable across diverse viruses and transgenes. Having the knowledge gained across diverse viruses and transgenes ensures that the new products that come into the company will still produce high titers. We realize that it's very easy on the back of the envelope calculation for significant cost savings of almost greater than 75% when you factor in all of these combinations of savings. Another avenue we're exploring is an evolution from transient transfection through to packaging and producer cell line technology in the context of AAV as well as in lentiviral vector development. The industry as a whole had suffered for a long time in the production gap. There was realized loss of titers when switching from adherent to suspension cultures. We've overcome this through the evolution of the 4G9. We consistently observe high titers. There's a significant advantage for having packaging cell line technology. The obvious cost savings attributed to the reduction in plasmids and transfection reagents, as well as the advantages of scale in perfusion culture. So high density cultures, lower volume. Given that we don't need to include complex transfection, allows us to lower the overall plasmid burden while taking advantage of increased density at scale. Viruses produced internally through packaging cell line technology tend to be better quality. They tend to produce more fulls, and they tend to be more uniform in distribution. So a lot less partials, a lot less empties. And for those customers that have programs using similar capsids, there's even significant cost savings. It's an early stage entry to justify a single platform using a single technology across multiple modalities, significantly reducing overall cost to the consumer. Of course, there are disadvantages for innovators. Generally, you need freedom to operate and accessibility to high production cell lines. And this early often is not the case for drug developers who are focused on clinical development of a gene therapy candidate. And so in that respect, the mindset is to just do transient transfection processes. And all the way through the clinic, that's been a successful strategy. However, as we think to the future, lowering the reduction in COGS and switching to packaging cell line technologies will be prudent. Bridging studies have often been difficult, but there's a requirement to show that gene therapies produced by different modalities produce the same gene therapy in the end. And this is typically done by clinical bridging studies. However, regulatory bodies are allowing speed to market and reduction in costs by using analytical bridging studies as a de-risking mechanism to show that the virus, by all biophysical and biochemical properties is identical. And it goes without saying that developing packaging cell line technology is not easy to do. It takes a huge investment up front. It's a very difficult technology. And in a race to be first to market with critical gene therapies and cure patients, innovators generally and typically do not invest in packaging cell lines. However, at SK, we are building this technology as an off-the-shelf offering. To start off with a typical VSVG lentiviral packaging cell line, as well as the standard offerings of AAV as discussed by Margie and AAV 12589 packaging. However, the technology is certainly amenable to any capsid and the evolution to a single cell packaging line. At SK PharmTecho, we truly believe we are a single source CDMO. We purpose built for this concept. We have early stage development. Typically, R&D stage is non-process. We control plasmids with plasmid accessibility. So as a single source, we can supply all your plasmid needs and sequences, along with very complex vectorology and plasmid engineering services, from cloning, triage, repair, identification of issues, ITR stability, cis plasmid issues as far as promoters and promoter design. And obviously, as we discussed, cell line development, using 4G9 to generate individual cell lines, inducible switch technology, and of course, customer-centric packaging cell lines. We also do small-scale R&D preps to save on costs, to produce high-quality, small-scale viral preps for cell-based assays or even in small-scale in vivo assays for mice. And of course, assay development, we can assist with supporting assay development for potency assays, release assays, etc. Because we leverage our internal R&D technology directly into our internal process development, our process development team through scale understands the nuances and especially how to work with 4G9, such that we've worked out a lot of the potential problems that could exist. And so in process development, from early stage DOE all the way up through 200-liter pilot engineering runs in bioreactors, we've observed performance of the line and know how to work with it. But also speed to market is in process testing and analytics. So there's no need to externalize testing, handoffs between CDMOs or other testing organizations. We can do in-process analytics in real time as samples are being made. This is a significant cost savings and obviously a significant reduction in time. And because we've done all of this together as one team, we've optimized this platform using the SKPT4G9 cell line. Follow that again under one roof is GMP manufacturing. We're a validated FDA compliant facility, audit ready. We've complete regulatory and quality service offering, and that starts from day one, from when an asset enters the building, all the way through commercial. Our regulatory and quality team works hand in hand with the R&D group, the process development group, and the GMP. So to ensure that all quality and assurance is met prior to filing, as well as IND and NDA application services. Along with the process testing and process development, there are also compendial GMP assay support to include all GMP release assays for product testing, microbiology services, as well as replication competent AAV and replication competent LVV assays. This ensures that customers really can leverage all the technology under one roof, seamless transitions and handoffs, and very quick speed to market, over a year of savings in time, without having to transfer between CDMOs, wait for results, transfer tech samples, etc. Just like to thank the folks who developed this line in the R&D group. Spent over 2 years in development. Myself and Margie, who spoke, and as well as all the wonderful scientists who also have done a lot of the upstream and downstream work to show performance of the line. Our associate scientists, Nan and Kelly, who work in upstream team, as well as our downstream scientists, Srivanya and Khan, who have done all the analytics and testing purification on 4G9. And lastly, I'd like to thank you for your time. If you have any questions, we're here for you. And please scan the barcode, and it'll directly link to the site and all the information that you need. Thank you.
Thank you so much, Marguerite and Brian, for a great presentation. We're now going to start our Q&A. So our first question is this cell line available for GMP vector manufacture? Whoever wants to take that one.
Margie? I'll answer it. The cell line, yes, it is available. It has been master cell banked, and it has been tested for all the adventitious agents. So the line is currently available. And then, of course, we would offer the creation of working cell banks. And those
Great.
working cell banks would be yours.
Thank you so much, Brian. Our next question is, what is the most important thing in packaging of lentiviruses beyond plasmid quality, cell line, and transfection method?
I can take that question. So lentivirus is generally a bit complicated. What we often see is while the titers look high, or what we're producing, sometimes it doesn't necessarily translate when you look at transducing units. And one of the things we didn't talk about, but one of the services that we do offer, and sort of both for customer centric, but as well as from an R&D perspective, is trying to understand a lot of the cryptic splicing that happens using very comprehensive and complex next-gen sequencing technologies. So really understanding where splicing events occur that lower your transgene expression. We also look at the sequence in general, look at GC-rich content, and try and minimize where those hotspots for splicing would occur. And so that's a core facility that we have at SK Pharmteco, and does NGS, MySeq, PacBio, and other comprehensive analyses.
That was a great answer. Thank you, Brian. We have some great questions coming in. Please feel free to send some more. Any we don't get to today, we will reply to by email. So our next question is, how does cell line stability affect the consistency and quality of viral vectors over multiple production cycles?
I can take that one. I think it's important for the cell line to be stable so that you have consistent, cell growth and consistent product coming out, vial thaw all the way to manufacturing stage. So for us, the main key was monoclonality allows for preventing and mitigating cell line drift, so that your cells are very comparable from thaw to manufacturing. And then at the same time, we've also done studies with extended passaging to monitor cell performance, cell growth, doubling time, and we've also done AAV serotype panels about every 10 passages, all the way out to about passage 60, to monitor titer and product to verify consistency.
Thank you, Marguerite. Our next question is, what are the key factors to consider when selecting a cell line?
I can take that too. I think that's also very tied into what I just mentioned about stability throughout, so consistency, and then, product quality. It's all very tied in. I think it's kind of this very similar answers to the previous question.
Again, I would also add, we giving careful consideration to just, obviously everybody wants production and the maximum production, but we've also given, thought to how the cell behaves and would behave in larger scale processes when you start scaling up to 200 liter. So rapid proliferation rates is not always significantly as important because generally what we see is that as cells grow faster, and if they're sort of refractory to the transfection process, and they continue to expand exponentially during that transfection process, the particles in the end tend to be a little bit not as clean. And so what we've noticed with 4G9 is that during transfection, the cells essentially slow down. They don't undergo significant doubling post-transfection. And we see a lot cleaner particles, as evidenced by the AUC and other orthogonal assays, such as mass spectrometry. We've also employed other strategies, although not discussed, but to look at enhancer technologies. And what we see with enhancer technologies, we can improve production even further than what we've seen around 10E11 VGs per mil. And in that case, the cells really stop growing entirely. So and if you look at the AAV literature as a whole, there's definitely a causal relationship between the cell cycle and proliferation, as well as, um, virus production.
Thank you both. That was a great answer. Our next question is, do you have an RCAV assay?
The answer is yes. And if you, I guess, visit that QR code, there's also a white paper that we have for RCAUV, and we'll be releasing an RCL, replication lentivirus white paper as well. And we do that in-house in our testing group, and that's been validated for obviously AAV1 and 2, but we can run it for all other serotypes. And
Excellent.
that's a cell-based assay coupled with dPCR.
Great. Thank you. Our next question is, how does the choice of cell line impact the efficiency and yield in viral vector manufacturing?
I can take this one too. As you've seen, and I think what segues into or parlays what I had just said before, is that you want to have a cell line that's stable, and we're measuring that using genomics approaches. We've shown it through functional assays that over, we extend the passage, and this is in the white paper, up to roughly 60 or 70, that we do not lose production. While that's not what usually happens in the manufacturing setting, it does suggest that the line is highly stable, that there isn't significant genetic drift from a production standpoint. You want to have optimal proliferation rates, because you need to grow cells up in a relatively quick amount of time. So we would like to have around a day, and that's exactly where we are, around 26 to 27 hours. So that allows for, and that's consistent. So what that does is allows the manufacturing team and commercial manufacturing, to build schedules that are consistent. And so there's a lean approach to, manufacturing, along with the ability to scale and produce high titers. So coupled, that's a significant cost savings.
Thank you so much, Brian. Next, we have, what strategies can be employed to enhance stability?
I think I'll take this again along the same lines. We've tested functionally stability, and we're looking now at very comprehensive NGS, RNA-seq, SNP analysis, whole exome sequencing to understand differences from the evolved line 4G9 versus parental. We're looking at differences between passage from very early passage through extended passage, and also as well as our MCB, to really understand what could be or may or may not be happening to the cell line, as well as interrogating the pathways that are hyper or hyporegulated. And then using that technology, we can employ a whole host of proteomics approaches to understand signaling, and as well as what pathways are activated or deactivated to improve performance. So that's kind of a pie in the sky answer. There's a lot that goes into that, and I think very few efforts to date across the whole landscape of cell and gene that have really started to comprehensively interrogate how different cell lines perform and link that back to pathways that affect that. We know that enhancer technology does interrogate certain pathways, and they do, have improved performance, but that's also can be highly cell specific. And so we're starting to do this work now.
Some great insights there. Thank you so much, Brian. Unfortunately, that is all the time we have left for questions today. Like I said, any questions we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so please look out for an email from us at the link. All that's left is to thank Marguerite and Brian once more for an excellent presentation, and thank you to the audience for listening. We hope you'll join us again soon